name is Saood and today is my presentation कल्चर मीडिया। तो फर्स्ट ऑफ ऑल हम करेंगे कि कल्चर मीडिया क्या होते हैं और हम क्यों इसको बनाते हैं। तो चलो स्टार्ट करते हैं। फर्स्ट ऑफ ऑल जो हमारे साथ कल्चर मीडिया होते हैं much of the study of microbiology depend on the ability to grow and maintain microorganism in the laboratory, and this is possible only if suitable culture media are available. A culture medium is a solid or liquid preparation used to grow, ट्रांसपोर्ट एंड स्टोर माइक्रोऑर्गेनिज्म। कल्चर मीडिया हम इसे बनाते हैं कि इसमें ये सॉलिड और लिक्विड का होते हैं और इसको हम यूज जो करते हैं वो ट्रांसपोर्ट के लिए एंड स्टोर माइक्रोऑर्गेनिज्म के लिए यूज़ करते हैं इनको। टू बी इफेक्टिव द मीडियम मस्ट कंटेन all the nutrient that the microorganism required for growth. Specialized media are essential for isolation, identification, antibiotic sensitivity, water and food analysis, industrial microbiology, and other activities. First of all, जो हमारे साथ होते हैं complex media that contains some ingredient of unknown chemical composition or complex media. Such media are very useful as a single complex medium may be sufficiently rich and complete to meet the nutritional requirement of many different microorganisms. To be effective, the medium must contain all the main type of culture media or basic, enriched, selective, indicator, transport, identification। जो हमारे पास डिफरेंट टाइप की जो मीडिया है उनमें एनरिच मीडिया, सेलेक्टिव मीडिया, इंडिकेटर, ट्रांसपोर्ट, आइडेंटिफिकेशन ये सब आते हैं। बेसिक मीडिया जो हमारे साथ होते हैं these are a simple media such as a nutrient agar and nutrient broth that will support the growth of microorganisms that do not have a special nutritional requirement. They are often used in the preparation of enriched media to maintain stock culture and control strain of bacteria and for subculturing pathogens from differential or selective media. Prior are performing biochemical and serological identification test। जो बेसिक मीडिया को हम यूज़ करते हैं वो सेरोलॉजिकल जो सेरोलॉजी का सेक्शन है उधर हम यूज़ करते हैं और बायोकेमिकल एंड सेरोलॉजिकल आइडेंटिफिकेशन के लिए हम यूज़ करते हैं जिसमें यूरिन टेस्ट होते हैं और ब्लड का भी कर सकते हैं। Enriched media are required for the growth of organism which exacting growth requirement such as influenza, Neisseria species and some streptococcus species. Basic media may be enriched with whole or lysed blood, serum peptone, yeast extracts, and vitamin and other growth factor. Selective media, these are solid media which contain substance, e.g. a bile salt or other chemical dye, antibiotic which inhibit the growth of one organism to allow the growth of another to be more clearly demonstrated. A selective media is used when culturing a specimen from a site having a normal microbial flora to prevent unwanted contaminant growing. A pathogen media made selective by incorporating antibiotics are usually expensive. Other way to select organism incubation condition may be used to select organism। इसकी जो एग्जांपल है वो एरोजिनोसा is inhibited by anaerobic condition। Indicator differential media, these media to which dye or other substance are added to the differentiate microorganism may differentiation media distinguish between bacteria by incorporating an indicator which change color when acid and produce following formation of specific carbohydrate। For example, McCon की agar Or many media used to isolate pathogen are both select or enrichment or both selective or differential. ट्रांसपोर्ट मीडिया हम इसलिए यूज़ करते हैं कि जो they are mostly semi-solid media that contain an ingredient to prevent the overgrowth of common cells and ensure the survival of aerobic and anaerobic pathogen when a specimen cannot be cultured immediately after collection. There is a particular important when they are transporting microbiology specimen from health center to the district microbiology laboratory or specimen the regional public health laboratory. Example of transport media include carry Blair medium for a carry Blair medium of preventing and MS transport medium for ensuring the visibility of gonococci. Identification media. These include media in which substrate or chemical are added to help identify bacteria isolated to primary culture. Example include peptone, water, sugar, urea, both are chemical, iron, agar. Organisms are mainly identified by a change in the color of the medium and or production of gas. Organism used to inoculate identification medium must be a first isolated in pure culture. Cost, availability and stability are different media to tropical countries. Training and experience of laboratory staff to preparing, using and quality controlling culture media.